Hello students, welcome to George Biology. We will start with ribosomes. Ribosomes, they are observed as a dense structures under the microscope by George Pallady. In the year of 1953. Now these members, they are non-membrane bound structures. They are non-membrane bound. They consist of RNA and proteins. They consist of RNA and proteins. As they consist of RNA and proteins, they are considered as RNP particles also. What does it mean? RNP, that means ribonucleoprotein particles. So ribosomes, they are also called as ribonucleoprotein particles. Why? Why? Because they consist of RNA plus protein composition. Now these ribosomes, they are of different types. It can be 70s type of ribosome. It can be 80s type of ribosome. Now if you look at the 70s type of ribosome, the 70s type of ribosome, it is observed in both prokaryotes as well as eukaryote. So in case of prokaryote, that is in cytoplasm, you can find it. In case of eukaryote, that is present in the mitochondria as well as plastids. Then what about 80s ribosomes? Now this 80s ribosomes, they are observed in the cytoplasm and connected to the endoplasmic reticulum of eukaryotes. So if a question is asked, what is the type of ribosome that is observed in both prokaryotes and eukaryotes? That is 70s type of ribosome. Either prokaryote or eukaryote. In both the cases, ribosomes, they are observed in the form of a dimer condition. It means that consists of both a larger subunit as well as a smaller subunit. So this is a larger subunit, that is 50s subunit, and a smaller subunit, that is 30s subunit. Now these two subunits, they are associated or dissociated depending on the presence of Mg+2. So if Mg+2 is present, now that will be united. Now if this Mg+2 is absent, now that will get disassociated. So here, if it is getting disassociated, that separates as a larger subunit and smaller subunit. And this larger subunit, it is made of specific proteins and rRNA subunits. So here, this is made of 34 protein molecules and it consists of 23s rRNA subunit and it consists of 5s rRNA subunit. Remember, this entire details that will be helpful for us in the molecular basis also. It means twice we are not going to study the ribosomes. Here itself we are completing it full and final. Now, coming to the smaller subunit. Now this smaller subunit, it is made of 21 protein molecules and it is made of 16s rRNA subunit. Now this is about the 70s ribosome. We'll go with 80s ribosome. Now here in case of 80s ribosome also, it is a dimer structure which consists of a larger subunit and a smaller subunit. And this larger subunit is a 60s subunit and a smaller subunit is a 40s subunit. Here also same point. The association is due to presence of Mg+2 and the dissociation is due to absence of Mg+2. Now, if the Mg+2 is absent, now that helps in formation of two subunits here, a larger subunit and a smaller subunit. The larger subunit, it is made of 40 protein molecules and it consists of 28s rRNA, 5.8s rRNA and 5s rRNA subunits that are present. 40s subunit. If you talk about the 40s subunit, now in this one, it is made of 33 protein molecules and 18S rRNA that is present. Now this is a property of 70S and 80S ribosome. Here we are representing the term of S. Now what does this mean? S that represents Swedberg's unit. Swedberg's unit, which is a representation of sedimentation coefficient. Sedimentation coefficient. Now what does this represents? It represents about the size and density of the ribosomes. Now here, how the distribution of RNA and protein that are taking place in 70S ribosomes and 80S ribosomes. If you talk about RNA and protein, in the case of 70S ribosome, RNA that is distributed as 60% and protein is comprising about 40%. At the same time, 80S ribosome, if you take, here it is quite opposite. 40% RNA, 60% protein distribution that is present in case of 70S as well as 80S ribosomes. Let's go with the function. Now if you talk about the functions, it helps in synthesis of proteins. This we already know. It helps in synthesis of proteins. Now what type of proteins that it will synthesize? Now if you talk about the synthesis of proteins, the endoplasmic reticulum connected ribosomes that helps in synthesis of secretory proteins. And this secretory proteins in further that helps in formation of transport proteins. It helps in formation of transport proteins or else tunnel proteins. Number 2, the free ribosomes which are present in the cytoplasm. Now that will help in formation of non-secretory proteins. And these non-secretory protein that helps in formation of enzymes and different structures inside the cell. Now these are the functions of ribosomes. Let's take 1 or 2 questions here which can be detailed in the exam. What is the type of rRNA subunit which is observed in both 70S ribosome and 80S ribosome? What is the answer? That is 5S rRNA subunit. Let's take one more question. Out of the following, which is odd with respect to the subunits of 80S ribosome? Number 1, 28S rRNA. Number 2, 5.8S rRNA. Number 3, 18S rRNA. Number 4, 16S rRNA. What is the answer? That is 16S rRNA. It's a property of prokaryotic or else 70S ribosomal subunit. Now this is all about the ribosomes. In the next session we'll go with cytoskeleton.